Greetings, everyone. My name is Joaquim Bruno, and I come from the Behavioral Biology Laboratory at the Federal University of Rio Grande do Norte, Brazil, to present to you my research, which is entitled, Does Environmental Complexity Affect the Behavior of the Ant Dinoponera quadriceps? My project addresses the concept of environmental enrichment and its applicability in the invertebrate animals kept in captivity. Environmental enrichment is defined as a set of processes that increase the complexity of the environment in which an animal is inserted. In addition to promoting well-being, environmental enrichment also prevents the appearance of stereotyped behaviors and encourages natural behaviors in animals kept in captivity. The benefits can go as far as to increase reproductive success in individuals. Although enrichment can occur in any of the 5 aspects listed here, this research project focuses only on one of them, physical enrichment. To do so, 2 colonies of the ant Dinoponera quadriceps that were kept in captivity were exposed to different levels of enrichment of their enclosure, as exemplified by the figure on the left. This 30-centimeter ant occurs throughout the northeast regions of Brazil and is part of the Ponerini subfamily. They do not have morphologically defined castes and constitute colonies of up to 50 individuals. The colonies are kept in a square meter enclosure, which includes 5 feeding dishes and 1 bigger container for water, besides a nest made of plaster with 6 chambers and a glass ceiling for observation that is being represented by this gray figure in the upper corner. During this research, the colonies were submitted over a month to 4 different levels of environmental complexity. At every new week, the materials were added to the enclosures as layers stacked upon one another, each configuration more complex than the previous one. Here you can see the configurations of the different treatments. The first one on the upper left corner is the control. The second one is the treatment where the substrate was added. The third one on the bottom left has rocks and pebbles, and the last one was added plant matter. Through our observations, it was possible to perceive changes in several behavior patterns of the colonies. Initially, it's possible to point out a significant increase in the number of individuals outside of the nest, roaming the foraging arena, a trend that repeats itself when we analyze the frequency of active foraging behavior. Other interactions behaviors with the environment vary between treatments, such as chemical marking. On other hand, inactivity was a behavior that showed a significant drop as environmental complexity increased. These results demonstrate how more complex environment can offer a greater variety of microhabitats to be explored, in addition to pointing out another issue. The lack of enrichment in the environment in which a model species is kept can bias the research results, as it does not allow for a natural repertoire of the animal behavior. I hope this research work illustrates how environmental enrichment can be valuable as a tool for studying social insects, these animals that have such a particular way of perceiving and interacting with the world. I would like to thank the team of my lab, especially my advisor, Professor Arrilton Araújo, and thanks to the ABS for the opportunity to showcase my work. Hope you all have a great meeting. Obrigado.